Hello, this is Dr. Dan Guerra coming to you from Authentic Biochemistry and VeraMed Studios. I'm giving a talk today called the Dialectical Paradigm for Brain Dysfunction Research. And I'm delivering this as an open symposium talk, and I want to get started right away because we have a lot of information to cover. So let's get started. All right. So, first of all, let's go back one step here and make sure that that's up there. There we go. All right, now from the beginning. All right, so let's get started. I am Dr. Dan Guerra. That's my email address, and I am the owner of Authentic Biochemistry and co-owner of VeraMed, two biomed consulting firms. That's my Facebook address when you need to contact me. That's how you're going to be able to do it. And any questions about this talk or any other talk that I give, I'd be really glad to discuss with you and also discuss what we do at VeraMed and Authentic Biochemistry. Authentic Biochemistry is also a podcast I do, and VeraMed is a regular lecture series that's presented on various platforms, including YouTube. All right, so I'm going to start off with a concept that I think science is often overlooked, and that is, what is the logical progression of our research? So one of the foundations of logic and attest whether or not there are truth statements that can be validated and found in sound arguments, two different things, of course, is whether or not they can pass a scrutiny of the square of opposition, which involves basically an interpretation of the categorical logic of the scientific paradigm to be discussed. That's what we're doing right now. So traditional square of opposition, you have a logical opposition, which occurs in most standard forms like this. They have to have the same subject and predicate terms, and they differ in quality or quantity or both. Also, consider the kinds of opposition that can arise with differing quantity and quality. They differ in both. They differ only in quality, but not quantity. They differ in only quantity and not quality. And the same quantity and quality, that's the trivial case, of course. So given the assumption made within classical, we'll call it Aristotelian categorical logic, that every category contains at least one member, the following relationships depicted on the square will hold. Let's go through them. Here's a square of opposition. You have A statements, E statements, I and O, which we're going to go through right now. We're going to now introduce our concept of neuropsychiatric disease. Okay, so firstly, A and O propositions are contradictory as are E and I propositions. Okay, so propositions are contradictory when the truth of one implies the falsehood of the other. Conversely, here we see the truth of a proposition of the form all S are P implies the falsity of the corresponding proposition of the form some S are not P. Okay, so let's take a look at this. For example, the proposition is that all neuropsychiatric disorders, which we're going to be calling ND, are modifiable, that is treatable. Okay, A is true. Then the proposition some NDs are not modifiable, O, you see across the square, the O proposition, right? That one must be false. Similarly, if no NDs are modifiable, that's the E proposition, the universal negative. If that's false, then the proposition some NDs are modifiable, or the contradictory across the square, right? The diagonal across the square must be true. Okay, so this is a way for us to get at examining how to develop a hypothetical deduction, right? When we're talking about something very broad and general, like what I'm introducing here, or something very specific, like talking about a neurotransmitter or a pharmaceutical therapy, for example, or surgery. Secondly, A and E propositions are contrary. So that is an important distinction between contrary and contradictory. Let's go through this. Propositions are contrary when they cannot both be true. An A proposition, for example, all neuropsychiatric diseases involve precise neural correlates, cannot be true at the same time as the corresponding E proposition, no NDs involve neural correlates. Okay? Now note, however, in this square of opposition, something emerges, that corresponding A and E propositions, while they are contrary, they do not contradict. So while they can't both be true, they can both be false, as with the examples of all NDs have unique psychiatric presentations and no NDs have unique psychiatric presentations. You see what I'm saying here? Contradictions versus contrary are two different distinct sets of premises. Okay. Next, let's examine the I and O propositions, which are sub-contrary. Propositions are sub-contrary when it is impossible for both to be false. Okay? Because, for example, detecting and diagnosing neuropsychiatric diseases could simultaneously delay the onset and progression of the disease is true. Failure to detect and diagnose NDs would not simultaneously delay an onset or progression of disease, has to be false because they contradict one another. Note, however, that it is possible for corresponding I and O propositions to both be true. That is, some NDs promote social and economic dysfunction, and some NDs do not promote dysfunction of the same category, social and economic. Again, I and O propositions are subcontrary, but they are not contrary, nor are they contradicting one another. So I and O propositions do not contradict. They are only subcontrary. Okay, so that means they can indeed both be true, whereas the contraries can both be false. See already how you're starting to develop a set of distinctions. All right, lastly, the two propositions are said to stand in relation of subaltern when the truth of the first, which is called a subaltern, implies the truth of the second subaltern, okay? Or, excuse me, the superaltern is the one that where you have the initial premise, and then implies the truth of the second, which is called a subaltern. So you have superaltern and subaltern, but not conversely. So you can't reverse the order. So A propositions stand in the subalternation relation with the corresponding I proposition. So that means the truth of the A proposition, for example, my example, all neuropsychiatric diseases are biochemical. I'm a biochemist. Implies the truth of, I added that, implies the truth of the proposition, some neuropsychiatric diseases are biochemical. Of course. However, the truth of the O proposition, some NDs are not biochemical, does not imply the truth of the E proposition, no NDs are biochemical. Of course, right? So in traditional logic, the truth of an A or an E proposition implies the truth of the corresponding I or O proposition, respectively. Consequently, the falsity of an I or an O proposition implies the falsity of the corresponding A or E proposition, respectively. Okay, so see how right away we can do a whole lot way before we even get to examining how we want to do a research project by putting it through the square of opposition, asking what is a logical association and what is not a logical association. So what's the resonating feature here? The truth of a particular proposition does not imply the truth of the corresponding universal proposition, nor does the falsity of a universal carry downwards to the respective particular. Now, there are a lot of philosophical and indeed I should say scientific paradigms that don't accept universality. Universality is only nominal, right? That is, it's only in name only. Or perhaps it is ideation. Maybe you can have an idea of a universal, but an actual universal in nature has often been argued against by various philosophical paradigms. I would think you'd want to imagine and reason that on your own to determine whether or not you think universals really do exist in your particular scientific discipline. Now, there's an existential import here. without the traditional presuppositions of existential import. That is, by that I mean the supposition that all categories have at least one member, then only the contradictory relation actually holds. On what is sometimes called the modern square of opposition, then the lines for contraries, subcontraries, and subalterns are all erased, leaving only diagonal lines for the contradictory relationship. So that can be a distillation of this square. And that might be the first thing you want to use when you're thinking about how this might fit around a boardroom or a lab meeting when you're discussing what kind of experiments to do. So if we can't presuppose and therefore assert as the first premise, because it's not demonstrated, Aristotelian term there, the concept that, for example, all neuropsychiatric diseases have definable and thus treatable biochemical correlates, then we cannot really assert that some NDs have definable and thus treatable biochemical correlates. Therefore, following along this logic, we can only say that neuropsychiatric diseases may or may not be biochemically correlated and thus therapeutically treatable. And I say QED to that. All right, let's take a look at a paper real quickly, and then we're going to go in and out of looking at manuscripts and follow through to get into my dia-event ontology paradigm. Here's a paper published about a year ago in Metabolomics. This is an example of what happens when you review the literature in neuropsychiatric disease, and then you try to compare it to other literature. Okay, so the project is to examine what's already published here and determine, looking at the whole corpus of that scientific literature, whether or not it fits in and whether or not we can find the correct way to determine if it fits in to the other paradigmatic statements associated with a specific research paradigm. So here we go. Schizophrenia is a complex brain dysfunction with occult pathogenic mechanisms. I think we all would agree with that. It involves impairments of motivation, cognition. There are no effective therapies, really. So there is no major drive to determine the endophenotype and its associated pathoneurobiological correlate. There's a lot of research in this area, but what exactly are we supposed to be looking for in terms of a biochemical endophenotype correlate? Now, there are candidate-associated genes, of course, and proteins that must be linked. Excessive loss of gray matter and also destruction of neuronal synaptic structure have been observed and suggested as being associated with schizophrenia. Now, there are a few candidate schizophrenic relevant proteins, including the RGS4, which is the regulator of G protein signaling 4, DISC1, which is disrupted in schizophrenia 1, SHANK3 or SH3, which actually is found as a domain in a lot of other proteins. And that is the multiple ankyrin repeat domains 3, that's what the H3 is for. All right. Now, metabolomic studies in schizophrenic patients suggest that glucoregulatory processes, proline metabolism, triacylglycerol metabolism, and associated lysophosphatidylcholine turnover are all also correlated. I'm a biochemist, so this is where I'm really interested. I like looking, of course, at genes and transcription factors. That's part of biochemistry as well. But when we start talking about metabolism, that's when I really get excited, because I want it, because of the foundations of every cell and every cellular system, every organism is going to be bioenergetics and signaling, which is all a biochemical wheelhouse. So plasma levels of both choline and ethanolamine plasmalogens, those are ether-linked lipids, phospholipids, were decreased by 23 to 45% in patients with schizophrenia. That was something that was discovered in this paper, doing a metabolomic study. So, that seems like quite a bit, right? 23 to 45%. The paper then is the next chapter, they suggest, on schizophrenic lipidomics, and with hopes to better describe the endolipidomic profiles during the disease onset, probably prodromal phase, and then throughout the progress of the disease. Now, results showed this. I'm just giving you examples. 47 lipids were examined. They belonged to 9 different lipid species. They were significantly different between the naive schizophrenic patients and healthy controls, where triacylglycerol, cholesterol ester, and sphingomyelin, those are all lipids, different types of lipids, by the way, were upregulated in naive schizophrenic patients, and plasminoyl phosphatidylcholine, that's a plasmalogen, plasminoyl PE, that's a phosphatidylethanolamin, CE, remember that's a cholesterol ester, phosphatidylcholine and lysophosphatidylcholine, missing one fatty acid in, for example, the 2 position, and lysophosphatidylethanolamin, same thing, no fatty acid in the 2 position of the glycerol backbone, and acyl carnitine, all those are downregulated. That's a lot of different lipids, okay, 9 different species. So what can we say about that? Well, let's go on. The results further say that 50 lipids belong to 9 lipid species were significantly downregulated after antipsychotics. So now there's 50, 47 for the naive. Now we're at 50 with those who are treated with antipsychotics. And those included some overlap. Glycerol esters, triacylglycerol, fatty acid, glycerol cerebrosides, lysophosphatidylcholine, etc. You see them there. Oh, and ceramide shows up too. Now, triacylglycerol and glycerol ester were upregulated in naive schizophrenic patients, and listen to this, downregulated after drug treatment. Now, the plasminoyl phosphatidylcholine is downregulated in naive schizophrenic patients where antipsychotics strengthened the decrease of the plasmalogen PC itself, and lyso-PC. Now, if you're a lipid biochemist, this is all really very interesting, but it doesn't really explain how these lipids are linked, because if you look at the pathways, which is, of course, what I do, it's my wheelhouse, sure, there are linkages to these pathways, but there isn't anything that associates, say, at the transcriptional level for the gene products, which are going to be the enzymes for the biosynthesis of all these lipids, because a lot of them are associated and wrapped up with diet and exercise and lifestyle. And again, you're looking at blood lipids. That's not what's happening in the tissue. So there's a lot of problem here because you're looking at various complex lipid structures in the serum. And you're saying, well, maybe these are good biomarkers because they differ between schizophrenic naive, schizophrenic treated with antipsychotics, and then actually healthy people. However, when you try to correlate those differences with understanding something about how they link together in a natural networked pathway system, right? In a systems biological, systems biochemical approach, there isn't anything rational that comes out of it. So it doesn't mean there isn't something rational. It just means that at maybe first and even second tier level of looking at the square of opposition, when you're trying to compare this logically, very difficult to be able to line up something that shows that you're on the right track, even for generating a new, say, research project. So let me tell you this, 4 patterns of this study I'm summarizing them here. Glycerol ester and triacylglycerol were upregulated in naive patients and downregulated by antipsychotics. Now, what am I saying? That's me in red. Too broad to determine pathways. Could be digestion, deposition of lipids, or even lipoprotein metabolism. That is quite a lot of activity that's not being measured or any way controlled in this experiment. Secondly, the plasminogens PC and lyso-PC were all downregulated naive and further downregulated in drug treatments. That would say plasminogen effect could be significant. Ester-linked lipids give no clear pathway insight. So maybe there's something with the plasminogens because they are unique lipids in the blood. One of them, platelet activating factor, for example, which is involved in stress responses and, of course, blood coagulation. But it's just not enough for us to make any definitive hypothetical deduction from. Third, some lipids were preferentially downregulated after drug treatment, ceramide, glucocerebroside, and just fatty acid, but not changed in naive patients. It's an interesting effect, I say, on cerebrosides, because those, of course, are neurolipids, but not enough detail to explain plasma origin of these neurolipids. So where is it coming from? Why is it in the plasma, right? Maybe you need to look at the cerebrospinal fluid and get an idea of how it could be circulating, but that was not done in this experiment. So finally, the plasminogen PE as phospholipids, the lyso-PE, and acyl carnitine were all up or down regulated in naive patients, but not altered by the pharmacotherapy. Of course, that's completely inconclusive and inconsistent. It can't tell us anything. So let's do a dialectic. Lipidomics may reveal associations between dysfunctional metabolism and schizophrenia. Okay, good premise. Or the antithesis, lipidomics may not reveal associations. We can clearly put that in the square of opposition and say that's a possibility, of course. So the results indicate that while there are trends, the experimental design was not sufficient to link differences with either disease state, severity, pharmacotherapy, or even resolution. So, and that wasn't a good paper. I'm not putting down the paper. I'm just saying when you look at published work and you try to be logical about it, and again, you are a biochemist, or let's say you're a cell biologist or a geneticist, and you apply your discipline to it, what you find is that there are no direct correlations that you can faithfully and, let's say, intellectually, with an intellectual fervor, nail down specific networks that you know may be affected in a disease state. Just not there. Now, what are my dia-event ontological implications here? And I'll get to this in a minute, what I'm talking about. How about if we design experiments where traceable dietary nutrients are monitored by serial blood draw from pre- and post-digestion and post-hepatic and extrahepatic trafficking of lipoproteins during, let's say, a pulse chase? Heavy isotope deuterium tracing could be performed sequentially among lipids, carbohydrates, and proteins. It's not toxic to humans. A combination of serum monitoring for all the free lipids that look interesting and the lipoprotein fractions could, coupled with a selective enzyme profile, you need that too. How do these various compounds end up in the serum, right? Could help to avoid high baseline variation and indeed the biggest problem, ambiguity. Also, of course, postmortem analysis of the liver, adipose, and central nervous system components of the brain in disordered patients versus healthy controls, and also disordered patients on pharmacotherapeutics, for example, could all be used then to retarget this lipidomic profile. Now, here's a general paradigm for epistemic discovery of neuropsychiatric events. The intersection of all living systems is, of course, the macrocosm, but it also appropriately describes the human body and overall stress imposed by things like the microbiome, invading pathogens, autoimmunity, cancer, autophagy, and senescence. You can also add in there apoptosis. The development, differentiation, and signal transduction cascade network, including neuronal action potentials and the endocrine mediation, also compose an opposing three-dimensional trigonal plane where the central element is the homeostasis of the existing individual. Indeed, learning and the accumulation of memory and, in fact, workable knowledge are all part of a massive internal interactome. I'm a biochemist, remember, that can be understood relative to advantage, vectorial control, by that I mean directed or agency, okay, and a constant attempt modality, right? Is this existing, and is it necessary or is it not necessary? Modal characteristics of that category. So brain disorders can arise from perturbations to any of the components of the system, and they can manifest at the pathophysiological and, of course, probably at the neuroimmunopsychological level. There is an interlocutory biological system that I've identified that permeates all those features describing the neuropsychiatric interactome and its axis, and that, of course, is the immune network event ontology, not substance ontology, but events, right? We're not talking about structures, we're talking about changing activity over time. So here are some paradigmatics of my dia-ontological concept. The immune system has two functions in human ontologics. First of all, there's the classical role as a cellular, molecular, constitutive defensive mechanism, coupled to an adaptive and learning offensive armamentarium, like, for example, natural killer cells. Secondly, an agentic individual, somatic physiological, and indeed neuropsychiatric processive event rational self. In other words, the developing, learning human being, human mind, human central nervous system, human motor neuronal system, such as a person who learns how to play the piano, or learns how to run a chainsaw, or learns how to drive, or learns how to sing. That's what I'm talking about. How do you develop the self from biochemical networking? And I'm suggesting that the immune system provides both of those potential mechanisms. Now, it is the latter function that I just mentioned there, the agentic individual one, which works through epigenetic mechanisms and from which purposefully emerges the existing individual. Purposefully, okay? This is a self-perpetuating neural network that can learn via attention and an ascent, that is to belief to discovery, to stress, and to function within the world. So that is to avoid, for example, things like pain and, of course, death. Such a biologically adaptive phenomenon is accomplished by a homologous recombination. So here I'm back to biochemical mechanisms of variable regions of both the immunoglobulin family and the T cell receptor, TCR, in concert, of course, with the regular chromatin remodeling, the histone code, these are all epigenetic phenomena, and both the acetylome and methylome of coherent DNA. And also let's include interfering RNA and even splicing mechanisms. So let me continue with my concept. Dialectical event-based ontological record is there. Is there a phenomenological distinction between mind and the body? There would be a physico-chemical axis of interaction. If there is one, if there's a phenomenal distinction between the two, there would have to be a physico-chemical axis of interaction. Okay, that's a premise. This axis might be the molecular and cellular, adaptive, immunological interactome. Serves to generate neural tracts according to developmental, endocrine, and peripheral stimuli while maintaining repair processes such as DNA repair in the central nervous system by using the complex interactions of the cells that we find in the CNS. Each cell is going to have a different role, and they're going to be interactive. For example, the dendrocytes, astrocytes, microglia, the resident immune cells, and of course the neurons themselves. According to this view, my view, neural networks are processes becoming eventual in nature as contrasted with the substance ontology. So this is a part of like a process philosophy point of view, process science, I would say. So the sequence and developing pattern of modifiable events appear as energetic waveform fields. That's actually what's happening at the physical chemical level, rather than particles of matter, much like chemical bonds, are actually waveform fields. They're constantly in dynamic interaction with each other and with the world of the phenomena. So what I say is there's an event ontology that reaches across the neural network of genetic and environmental interaction and is articulated and synthesized through epigenetic modifications, the ones I just mentioned, of the classical immune system. So that's where the epigenetics is functioning on in order to have a communication network, thus creating a responsive plastic and elastic memory field capable of learning, ideation, imagination, and understanding, that is conceptualization, through time. So therefore I say this dialectically, thesis, antithesis, synthesis, arrived event ontology involves the central existing individual, agentically interacting through the present perfect just happened process, obtained across the inherited genome, the changing environment, and the transcendental working above both planes of the immunoepigenome through time. So there's the diavenome that I've published on. You've got the genome, the environment, and the immunoepigenome. So I say that there are neural networks and they are processes becoming eventual in nature as opposed to substantial. And I say the event ontology that reaches across the neural network, right, as genetic environmental interactions articulated and synthesized through epigenetic modifications of the classical immune system. Okay, and coupled with that, you have responsive plastic and elastic memory, what I just mentioned to you, and how that's very similar to chemical bonds. Chemical bonds are not stationary, fixed, static phenomena. So this does not violate any understanding of chemistry or physics. And I say the central individual agentically interacts through the events obtained across the inherited genome, the changing environment, and the transcendental working above this of the immunoepigenome through time. So what is event ontology? Dialectical event ontology is what I mean by that. It's an adaptational, processive, scientific accounting of physiological, rational human condition through cellular and molecular event ontology. That's my definition. That is my new paradigm. It's biological mechanism of action, linking pathopsychological states, for example, like Alzheimer's disease, to biochemical pathways, incorporates the immunoepigenomic induction of neural, endocrine, metabolic response, including bioenergetics, by the way, to the micro and macro environment through classical constitutive surveillance, this immune system here, and acquired, that's the second tier of the immune system, effector cellular humoral defense stratagems using reversible covalent modification, hydrophobic interactions of nucleic acids, proteins, and lipids. All the molecules are indeed involved. So die-event ontology offers a general molecular system theory for free will-driven, agency-based individual adaptation and a knowledge-acquiring physiological rational mechanism that better explains the core event ontology of human existence, including what we're interested in, health and well-being, we as medical scientists. Now applying my die-eventological paradigm to Alzheimer's disease. So this is the second run at this, so I call it the Alzheimer's disease die-eventologic Roman numeral 2. Take a look. The genome has various genes which we know are associated with Alzheimer's disease, right? The amyloid precursor protein, the neutral sphingomyelinase, the ceramide synthase. These are enzymes in lipid metabolism. I'm a lipid biochemist, by the way. The beta and gamma secretases, presenilin, tau protein, ApoE4, which traffics via the HDL, actually lipoprotein, and the so-called XC pathway, which involves cysteine and the synthesis actually of glutathione, therefore the removal of reactive oxygen, which can damage neural networks. What else is in the genome, which is more opaque and occult, but we know is there, are things like overall intelligence, adipose equipoise, like whether or not a person puts on a lot of, for example, visceral fat, and also maybe gender's involved. We just don't know because we don't have enough studies. So those are the genomic components. What's the environment for Alzheimer's disease? We know aging is involved. We know inflammation, recurring inflammation. We know that cardiovascular disease can be associated with developing Alzheimer's disease. We know infection, stress, a sedentary lifestyle, and that's all associated with a tremendous problem in worldwide, obesity, right? It's a epidemic. Education, that's an environmental aspect that the agent goes through. Intellectual avoidance, people that don't try to exercise their intellect tend to be people that have earlier onset Alzheimer's disease and get more severe earlier on and die from it. Any kind of alcohol or drug use seems to negatively impact, that is to promote Alzheimer's disease progression, both the prodromal phase and when it's fully on. Glutamate, the ingestion, utilization of glutamate, that's an environmental aspect. The iron, ferrous iron ferritasis pathway, which I've talked about in various lectures, which I don't have time to talk about right now, that involves oxidative metabolism associated with iron in the blood and in tissue. And then the MPTP, which is a transfer protein, which I don't have time to talk about. Those are all environmental aspects, however, not genomic. These are turned on in real time. Finally, what's about the immunoepigenome? Microglial activation, remember those are resident immune cells. Astrocyte dysfunction, well published in the literature. Oligodendrocyte malfunction, which is different than dysfunction. Malfunction just means it's doing something different than it's programmed to do at a given time space. Again, a die event. B, plasma cell recruitment, T cell differentiation and modulation. These have a lot to do with homologous recombination and homologous end joining. Cytokine, chemokine growth factor modulation coming from different cell types. That's all part of the immune system. And also, of course, it happens from epithelia and endothelial cells. Oxyphospholipids, once you make an oxygenated, after oxidizing a phospholipid, those act as their own immunoepigenomic targets and agents. And then what else? Of course, microRNAs, sirtuins, and histone acetyltransferases. Sirtuins tend to function as deacetylases or HDACs. And then, of course, DNA methyltransferases and DNA methylases. Removal of the methyl group also affects chromatin. These are all canonical epigenomic pathways. Okay? And this is what we've already discussed. You've got universal affirmative, no harm to individual, maximum benefit, rare occasional epistasis, gene-gene interaction that is. The universal negation. See, I'm bringing back now the square of opposition. Universal negation, severe harm to the individual, 100% interaction of the genes. Particular affirmative, some benefit to the individual, disinterested epistasis, meaning that whatever's going on with the gene-gene interaction doesn't seem to be networked. or that is coherent. And then you have particular negation, some harm to the individual, and you have a predicated gene-gene interaction or gene-product interaction. So this is just me superimposing the square of opposition to this paradigmatic re-understanding of a current, very important neuropsychiatric disease, Alzheimer's. And here's adding in now the specific flavors of what we know about it. Now, here's another way of looking at it. This is a step gradient looking at general anxiety disorder. And I published a paper on this recently, so this is from that paper. In utero development may be associated with general anxiety disease preconditioning. What will that utilize? Genetics, overall, the maternal environment, the biological environment, epigenetic coding during in utero development, and the fetal immune response to potential antigens. The next phase is parturition. There's a tremendous effect once exactly parturition occurs, according to the carbon dioxide oxygen balance, which can greatly affect neuropsychiatry subsequent to the birthing process. And that involves also, indeed, interestingly, biofuel, whether or not you beta oxidize fatty acids or you utilize glucose. Right at parturition has a tremendous effect on neural networking and on brain recovery during that very stressful parturition process. And of course, any local inflammation that occurs. Second, infancy and early childhood. Of course, now you're getting into nutrition, immune challenge, all of the antigens out in the environment, and microorganisms, of course, some of which are pathogenic. Genomic mutations that are going to be occurring throughout these early stages once the environment starts kicking in. Alternate splicing of all the RNA. Epigenetic programming, all the paradigms that we talked about, methylation, acetylation, ubiquitinylation, sumoylation, and then removal of all those compounds, even phosphorylation. And then, of course, indeed, neurological development is going on at the same time, right? There's all kinds of neural networking and rewiring and repruning and pruning. And this is all during what I call the GAD induction, right? General anxiety disease induction process. Finally, we have instantiation of GAD, full-blown. And this is from childhood to puberty to adulthood. Nutrition and exercise, again, playing a major role. Hormonal switching, right, during, for example, the pubescent phase. And then full-on neuroimmunoepigenomics is occurring during the entire life of the human. And that is going to be what I call full-blown GAD instantiation. So, what does verifying literature require concerning this immunoepigenetic tailoring of individual biomedical events? So here's a paper that was published some years ago in prostaglandins, leukotrienes, essential fatty acids. Yes, indeed, there is such a journal. Yes, indeed, I read it. Now, what is this paper asking? Is there a link between dietary omega-3 fatty acids? I know you've all heard of those. Vitamin B12, probably have heard of that. Certainly you've heard of folic acid. And it may involve bioenergetics and membrane-mediated, look at this, DNA methylation patterns. So this is a huge question being asked in this paper. Pregnant maternal intake or metabolism of any of those nutrients and fetal metabolism associated with it may suggest a role for maternal omega-3 fatty acid and vitamin B12 on, look at this, phosphatidylethanolamin N-methyltransferase activity. That's on the way to making phosphatidylcholine, as I'm sure you all know. Peroxisome proliferator activated receptor, PPAR, alpha or gamma, either one. Adiponectin, that's of course an adipokine. Signaling pathways, and both epigenetic processes like chromatin methylation and acetylation. All right? Now, you also can discuss AMP kinase phosphorylation of the DNA methyltransferase 1. That tends to decrease the methylation of promoters for several mitochondrial biogenesis transcription factors. So you phosphorylate that methyltransferase, you decrease the methylation of promoters, and those are all associated with several mitochondrial biogenesis transcription factors. That was found, published in the International Journal of Biological Sciences. It's another component because AMP kinase is regulated by lipid metabolism. Finally, ADO-MET is, of course, the methylation agent, right? Adenosine. It can be diverted to phosphatidylcholine synthesis from PE, thus altering membrane biophysical predicates, including those associated with mitochondria, the peroxisomes, and the plasma membrane. I know there's a lot of biochemistry. This is all just in one question. When you verify literature and does it according to immunoepigenetic tailoring of individual biomedical events, you find it everywhere. All right? So there's a competition, for example, for this adenosine between membrane retailoring And that has to do with the synthesis of phospholipids. And of course, DNA histomethylation, because that's the methylating agent for that. Remember, when you methylate promoters, you tend to decrease gene expression. And that, of course, is going to affect the immunoepigenome of the immune cells and of all the other target cells that the immune cells affect via chemokines and cytokines and growth factors. Here's another paper. We're almost done here. Here's a paper that's looking at acyl chain symmetry and polyunsaturated of brain phospholipids, which facilitate membrane vesiculation without leakage. Okay, here's some discussion. Phospholipids with two polyunsaturated fatty acids. I know you've heard saturated fatty acids versus mono versus polyunsaturated fatty acids in the diet, for example. Those phospholipids with two PUFAs make membranes prone to vesiculation, but highly permeable. So they vesiculate, okay, they form internal vesicles, and that makes them highly permeable. Asymmetric, SN1 saturated and having the two position of the glycerol molecule, that's the stereospecificity around the carbon atom of the glycerol molecule, having a polyunsaturated in the two position, which is a very common phospholipid, provides the trade-off between efficient membrane vesiculation and low membrane permeability. Isn't it interesting that most of the phospholipids have that kind of molecular geometry? Saturated in the first carbon, polyunsaturated in the second carbon. Very, very common. In fact, that number 2 fatty acid is often arachidonate or eicosapentaenoate or even docosahexaenoate, particularly in the CNS. Now, in incorporating the phospholipids, DHA, which is, of course, an omega-3 fatty acid, makes membranes more deformable than arachidonic acid. That's another thing that happens. Membrane being more deformable means that you can alter signal transduction cascades across that membrane, particularly, let's say, a synaptic associated membrane. So what they found is fatty acid profiles in specific molecular species are associated with membrane organization, such that phosphatidylcholine fatty acid are distinct from either phosphatidylethanolalamine or phosphatidylserine. So ADO-MET, adenosine methionine utilization, because that's what's used to make PC from those other phospholipids, those methyl groups, and thus competition with histone lysines, which become methylated, right, is in part, and those lysines are associated again with histone 3 usually, is in part regulated by diet and genetics. Environment and genetics dictating molecular species fatty acyl content. Okay, we're going to skip this one here. This has to do with hypomethylation. It's more epigenetics. Okay, and if I had time, I would talk about it. We're going to skip that. We're also going to skip the natural killer cell dialectical conclusion that I wanted to get back to from the very beginning of the talk, the metabolomics. I'm going to pass over this for time. I'm going to give you the final home run here. Dialectical perspective. Now we're talking about natural killer cells. I know I didn't introduce them before, but those are immune cells. Natural killer cells crosstalk with both innate and acquired immune cells, and thus they potentiate a signal that can transduce across multiple layers of signaling valence. Remember that I'm talking about the dialectical paradigm. Using a methylation patterning NK can be flipped to be agents of cytotoxicity or indeed inflammation, just by methylating certain proteins in NKs. The inflammatory associated output of natural killer cells involves cells of innate and acquired responding lineage. So gliomas, I know we all are aware of these, are characterized by low cytokine and chemokine secretion, and that leads to tumor growth via microenvironmental immunosuppression from these microglia, which can be overturned by infiltrating natural killer cells, which secrete these pro-inflammatory cytokines like interferon gamma, interleukin 15. That can trigger an anti-tumor myeloid response. That was published a couple years ago in eLife. Now see how that works. See how when I can look at the literature now with a different lens, the dialectical lens, I start seeing how things come together. What about chromatin instability or CIN? Very important in disease. It's linked to tumor metastasis via a mechanism that starts with an incomplete chromosomal sorting at actually interphase. And that generates many chromosomes. The instability generates many chromosomes that actually leave the nucleus. And upon rupture of their membrane, release their double-stranded DNA content into the cytoplasm of the cell. That will trigger an inflammatory response that taxis the cells, which starts metastasis. Now look at that, okay? Now how does that work biochemically? Again, It's associated with interactions in the chromatin, such as epigenetic mechanisms which can be induced by the immune system and the environment working together. So, chromatin instability phenomenon employs similar mechanisms to those associated directly just with natural killer cell inflammatory activity, suggesting a cellular transformation to dia-eventological pathways. So rather than just talk about cellular changes, now we're starting to look at it at a higher elevation. We're starting to see these are actually dia-event-ontological pathways that are occurring. And that's how I'm suggesting we need to start designing experiments at that higher elevation level. Therefore, the immunoepigenetic component of the trigonal planar dia-eventome, right, that interactive stage, can be obtained not only within myeloid cell populations, but indeed across all dividing cell lineages. And that can establish a processive and perpetual biochemical event ontology, which is going to be unique to the individual all the way to the final death. So here I put together what can happen in terms of autism spectrum disorder. You have pathogens and xenobiotics and antigens that cause molecular stress. If there's a failure to respond, there's a failure to elicit signaling. It ends there, okay? These are damaged molecular patterns, and these, of course, are toll-like receptors where molecular stress can also function. If that turns on, you can turn on the APC pathway. The APC signaling pathway can turn on an innate resolution, which involves intracellular phagocytosis or even apoptosis. Or the APC signaling pathway can turn on the lymphocyte activation and differentiation system. It can also then regulate directly inflammation, which can resolve the damage, or the inflammation can lead to DNA repair, okay, DNA damage repair. That DNA damage repair can induce mutations such as copy number variations, splice variations, and single nucleotide polymorphisms. And that, in conjunction with all this immune pathway response, leads to autism spectrum disorder. Now, this is a pathway paradigm using my dia-eventological perspective. So again, I published this recently in a review article I did on autism spectrum disorder. And you can read all of this. You can play this back and you can take a look at in more details of what I'm talking about. But you get the idea. The pathway has the ability to fix and repair. It can, for one thing, it can fail to respond, or it can fail to elicit a signaling. These are two different mechanisms. Or it can go through all of this signaling and inflammation, but it can resolve. Or in the inappropriate pathophysiological response, it can turn on certain DNA damage repair mechanisms, which will actually induce mutations, copy number variations in specific genes that, when their copy number is changed, will then cause a sequence of events, a processive sequence of events that leads to a multigenic, multicellular, overall physiological, genomic, environmental interaction, which involves the immunoepigenome, that leads to what we call autism spectrum disorder. All right, so let's wrap up here. Put me back on just to say goodbye. Hopefully that was, I know, a rather fast-paced talk, where I wanted to give you the overview of my dia-event ontological paradigm. I also wanted to lead you from the beginning where I started it by thinking about logical experiments, first logical experiments that you do in your mind, or you can do on paper, to be able to examine whether or not a given research pathway is one that's governed by truth preserving. If you can preserve what's already known at the level of the truth preserving, you have a sound argument to follow along and do a hypothetical deduction leading to a research experimental procedure that will give you, because you have the correct controls, etc., a more weighted and indeed a more infinitely penetrating understanding of the research you're doing. So what am I saying in general? We need to read the scientific literature, we as scientists do, much more than we do. We need to examine the papers. We need to slow down our pace. We need to look at the corpus of literature and not just what was published in 2018, 2019, but even something that was published in the 1990s and 1980s. A lot of research is left by the wayside, and there's no reason for it to be. Just because it hasn't been carried through because it doesn't get cited often doesn't mean that research and the data, the evidence, once verified, can have a lot of meaning to what we're doing in 2019 and beyond. That means we have to actually spend the time to read. We as scientists, we can't delegate to postdocs. We can't delegate to graduate students. We certainly can't delegate to technicians or lay people. We have to be the people reading the literature, which means the more time we spend on that, of course, maybe the slower the pace of our experiments because we think before we do them. I would argue that we can save money. We can also maybe save animal colony costs because we have less animals. Because my approach ultimately is to sure use the animal model where it's necessary, for example, to look at new drugs or new surgeries or other new kinds of therapies, but to move away from animal models and go directly to humans by sophisticating the scanning that we can do and by using things like deuterated compounds, which are benign to the system, and then do a more thorough omics research on how that deuterium is spread around doing GC mass spec or LC or LC mass spec differentiation of different plasma components and even indeed tissue biopsies. So I'm going to end here. Again, I'm Dr. Dan Guerra. That's me down there. This is VerevMed and Authentic Biochemistry. Those are my companies. There's the email address for VerevMed. I don't have one yet for Authentic Biochemistry. It's a podcast you can pick up on iTunes. I think I've got maybe only 8 or 9 episodes out there, but I'm working on getting a lot more. There's the website for VerevMed, and what VerevMed is a scientist verifying published research in medical biosciences, okay, evidence in medical biosciences. I guess I should remember what my VerevMed stands for. There's my Facebook page. Please go to that. There's a lot of information there on what I specifically do and the company I co-own, VerevMed, does and what Authentic Biochemistry has been doing. Here's my individual email. I know it's not the best idea to give out your individual email, but I do that because I really would like feedback on anybody who would be interested in discussing this at a consultancy level to be able to think about how we can use Ontologics to better get at neuropsychiatric diseases. So I'm going to end this talk like I do in all of my video lectures and all my live lectures, and thank you very much for the attention you've given me, by saying my sign-off, which of course is bye.